Stress can come in many forms on the farm, from travel to heat and chilling, from social disturbances, ammonia, lighting. There are a lot of experiences birds need to adapt to during a production cycle. Long-term stress or repeated stress will result in the body creating the stress hormone corticosterone, which can have a lot of negative impacts to the body, and some of which include suppressed immune function, decreased muscle mass, and enlarged liver size. My research has been focusing on how stress can change the bacteria in the gut, and if this can influence a bird's susceptibility to necrotic enteritis caused by the bacterium Clostridium perfringens. My studies have shown that stress can lead to there being less types of bacteria and also can change which types of bacteria dominate in the intestine. Importantly, stress has shown to increase the levels of Clostridium perfringens in the small intestine. In fact, when birds were fed the stress hormone corticosterone in their diet and exposed to the bacterium Clostridium perfringens, 80% of these birds went on to develop necrotic enteritis. In our latest study, we took intestinal contents from mature broiler breeders to create a complex mixture of bacteria that was administered to half the birds in the study the day after hatch, while the other birds in the study didn't receive this bacterial mixture. After 10 days, the birds were exposed to both stress and Clostridium perfringens. We found the birds receiving this complex mixture of bacteria remained healthy, while the other birds that didn't receive this mixture went on to develop necrotic enteritis, with about 75% of birds developing disease. So how is disease being prevented? There are a lot of complicated explanations for this, but this is a simplified one. So in the intestine, there are different zones or environments that each type of bacteria will live in and declare it as their own. When lots of different kinds of bacteria are present and a pathogen comes along, such as Clostridium perfringens, it's less likely to stick around when all the other bacteria are claiming these environments and the nutrients in them. In a stress intestine, though, where there are less types of bacteria, this leaves more gaps for pathogens to come in and take up residence. So when Clostridium perfringens comes around this time, it can overtake the intestine and lead to disease. For future applications, we're looking to see if there are any kind of specific bacteria that can help prevent necrotic enteritis, but we're also interested in looking at how the environment in the intestine is changing when birds resist disease to see if this is something we can select for through either diet or supplements. Thank you for listening. This research was done by myself, Sarah Zaytsoff. I'm a PhD student at the University of Alberta. All this research was completed at Agriculture and Agri-Food Canada in Lethbridge under the supervision of Dr. Douglas Ingalls. Thank you.
Thank you, Sarah, for sharing that synopsis of your research work. Remember, if you have questions for Sarah on the project, you can just ask them through the Q&A button at the bottom of your screen. As we take a look at your research, how did you know the mixture of bacteria administered to the birds was free of pathogens? How could you guarantee it was all kind of, I guess, good bacteria?
Yeah, we did a couple different things. So I did do DNA sequencing of it to check for there not being any other bacterial pathogens. We also sent it to some collaborators out in Eastern Canada to check for viruses, which are also really important to not have around. And then we also did isolation for parasites and stuff to make sure there were no lurking Eimeria species around.
So you made sure it was the good stuff, and then you were able to get pure results, sort of a thing.
Yeah, exactly. We also kind of used, it's kind of like an artificial intestine system. We use it's called a bioreactor in a anaerobic chamber where it's devoid of oxygen. So we were able to kind of grow it in there and kind of standardize it to make sure all the birds were getting kind of the same mixture too.
Okay, what about your research work? Has it been able to identify any particular bacteria that could help, prevent birds from acquiring pathogens? Are there any superstars out there?
There might be. I have just started doing the analysis on that. It's kind of the last thing that I need to get done. And there does seem to be about 3 or 4 different kinds of bacteria that colonize the small intestine that seem to really increase in those birds that were getting this complex bacteria that really reduce that densities of Clostridium perfringens And so the next step is kind of figuring out why that's happening. Are they interacting? Are they somehow metabolizing nutrients in the environment? So I'm doing what's called metabolomics analysis next. It's been completed and it has shown that the environments are completely different in the birds that got disease and the birds that didn't get disease. So it's just about figuring out which exact nutrients and metabolites are really there that are kind of helping out with that.
So it sounds like a lot of work yet to go, but the end game could be, offering some alternatives for this disease prevention then.
Yeah. I'm also looking at immune function. So I'm also trying to see if all these different bacteria can maybe stimulate the immune system in a positive way that could also help disease resistance.